Hi all, today I'm going to start with the biology session for kingdom Animalia. In that I will be doing a short video on Phylum Porifera, the first phyla that is coming under kingdom Animalia. Hope you know the general characters of Animalia. So according to biological classification, you know that the eukaryotes, which are not having any cell wall, which is having multicellular structure. Cell wall is absent, multicellular structure, and the levels of organization, if you are taking, you can see they will be having tissue level, or organ system level, even cellular level is there. So these organisms which are heterotrophic and without a cell wall and which is having a eukaryotic cell is placed under kingdom Animalia, and further classification depends upon the symmetry, coelom, segmentation, as well as the triploblastic, diploblastic germ layers. Based upon that only further classification is done. So in that, the first placed in animal kingdom is Porifera. So let's have a discussion about the phylum Porifera coming under kingdom Animalia. So look at my drawings and the videos here. So look at here, Porifera, which is commonly known as sponges. And if you take the habitat, you can see it is found in the marine ecosystem. And a few members you can see they are found in freshwater also, like the Spongilla. Spongilla diagram is given in NCERT. You know that it's found in freshwater, whereas Cycon, Euspongia, all that if you are taking, you can see it's found in marine ecosystem. And coming to the structure, you should understand it is the lower animal. It will be having cellular level organization. So out of all the phyla if you are taking, you can see sponges are the one which is having cellular level organization. And if you take the symmetry, you should understand it's having asymmetry. If you try to cut it into two equal halves through the central axis, you cannot cut them in any plane. So I should tell it is asymmetrical. Out of all the phyla that you are learning, you can see the sponges are asymmetrical through the central axis. If you try to cut them into two equal halves in any radial plane, you may not be able to get an equal right and left half. So I should tell it's an asymmetrical organism, and it is cellular level organization, and it's diploblastic. Two germ layers are also present in them. So you look at the diagram in that cycon. If I try to cut through the central axis in any radial plane, I may not be able to cut them into two equal halves. Euspongia also, Spongilla also. So that is why I am telling it's a asymmetrical organism which is having a cellular level organization, and it's diploblastic. If you take the germ layers, you can see there are two germ layers coming in the sponges and all. And body cavity, if you take, there is no body cavity, it's an xylomate organism. Now let me draw a diagram in order to explain the structures further. So you just look at the diagram which I am going to present here. If you take the body of the organism, if the sponges, you can see like what is seen in the picture, so many holes will be coming all over the body. That holes that are coming all over the body in the sponges, that is actually called as ostia. So there is ostia, small pores all over the body, that is called as ostia. So through that ostia only water will be entering into the sponges, into the spongocoel or in the cavity of sponges. So spongocoel is actually present at the center, and through to the spongocoel the water will be entering into the sponges through this minute openings throughout the body, that is actually called as ostia. Hope you understood this much regarding the sponges. And on the top there is again so many holes that is coming through which the water will be exiting out in sponges, that is called as osculum. Hope you understood the ostia and osculum that is responsible for the water canal system that is found in sponges. So this is the water canal system in which you can see the water enters into the spongocoel of sponges through the ostia and will be exiting out through the osculum. Now the endoskeleton, if you are taking in sponges, you should understand it will be having spongin fibers, or we can call it as spicules. Spongin fibers or spicules will be actually forming the endoskeleton in case of the sponges. So this much structure, hope you have understood in sponges. And inside that you can see they have lining cells also. Lining cells, you can see they have choanocytes, so collar cells will be there, which is having a flagella-like structure. Due to that lashing movement only, the water is actually entering and the water is pushed out of the sponges. That is also a point you should understand from this structure. So let me draw that choanocyte or collar cell that is coming inside. So choanocyte or collar cells also I am putting inside. 2-3 cells I will be drawing with red color. So in that you should understand they will be associated with the movement of water, as well as the inner cells also associated with gamete formation. So in sponges, the male and female gametes will be formed inside the spongocoel only. So male gamete and female gamete will be formed by the inner lining cell that also takes place in the spongocoel. So digestion may be intracellular. So hope you have understood up to here. So hope you have understood the sponges have small holes all over the body that is called ostia, and on the top there is osculum, and it will be responsible for the water canal system. Inner lining the endoskeleton is having the spicules or spongin fiber, and this inner cavity is the spongocoel. Lining also contains special cells that are actually called the collar cells or choanocytes. Hope you got this much. So the water movement in the water canal system I am showing in the diagram. In that you can see it's entering through the ostia and exiting through the osculum. Like that is the entry and exit. So what it helps in the water canal system. So from outside the nutrients, oxygen, all these are actually coming into the spongocoel and the lining cell through this ostia only through the water canal system. And the waste particle, the larva, the gemmules, the internal buds, etc., that will be exiting out through the osculum also because of this water canal system. So water canal system is actually found in the sponges. So functions of the water canal system I am mentioning in the diagram. Please note that the water canal system will be having the nutrients as well as the oxygen that will be entering. So oxygen and nutrients enter through the water canal system and the water, and it will be supplying to all the cells lining in the spongocoel. And after that you can see the exiting is the waste particle like carbon dioxide and other waste particles, the debris, etc. And even you can see the internal bud, the endogenous buds that are formed inside the sponges, they are called gemmules. They will be exiting through the osculum when this water is going out. So this much is the function of water canal system, which you can expect for the exams and all. What is water canal system? What is the function? In which organism it is found, etc. In CBSE exams you can expect. So I'm also putting some gemmules diagram. If it's a rough diagram, you should understand it's coming inside the sponges. They are internal buds. Now, next, after the gemmules. So gemmules are endogenous buds. You should understand budding two types are there. In yeast and all, it is exogenous bud coming in porifera. In sponges, you can see it is endogenous buds, that is gemmules. So hope you understood this much. Now, inside only they will be forming the male and female gamete. The male and female gamete is formed inside the spongocoel. Fertilization also takes place inside the spongocoel. So for easily remembering, I am also drawing that diagram. So let's have a conclusion about the symmetry. A symmetric organism, you cannot divide them through the central axis into two equal halves in many planes. So in my diagram, I have drawn only a part of the sponge, branches will be there. So it's a symmetric cellular level organization without a body cavity. So it's a coelomate. And it is diploblastic. These are the general points you should remember. Then coming to the structure, you should remember the ostia, the osculum, the spicules of spongin fiber, and there is also choanocytes or collar cell. And here there is male and female gamete that is formed inside, which is followed by fertilization to form the zygote. The zygote will further divide to form larva. So development in sponges, I should tell, it is indirect through larval stage. So after fertilization of male and female gamete, it will result in the formation of a zygote. The zygote will undergo further division and turn into larva, the embryo, and the larva will be formed. Once the larva is formed, you should understand the larva will be exiting. The diagram, the zygote that is going to turn into the larva, and the larva further development that is taking place outside the spongocoel after exiting through the osculum. So fertilization is internal, that you keep in mind. Development is external. The larva as well as the new porifera and all, the development will be external. So I'm just adding that also in the diagram. The zygote that is actually forming the larva, and the larva is turning into the new organism. So from the diagram itself, you can learn many points. That is the purpose of drawing all these diagrams. Hope you got this much. So hope you can see the larva that is undergoing changes and it's turning into the sponges. So I have drawn a diagram of cycle, just imagine like that, which is having the asymmetric structure and all, ostia, osculum, etc. I added in the newly formed member also. Then example part also very important when you are learning. Learn the 3 examples correctly. Cycon, Euspongia and Spongilla. In that Cyconoscypha, that is marine. Spongilla, the freshwater sponge, Euspongia, the bark sponge, that is also marine. So this much only is given in NCERT about phylum Porifera, the first phyla that is placed under animal kingdom. Hope you understood. So for classes like this, you can subscribe to my channel KDS Biomentor for NEET and CBSE.